Good morning, everybody. That's a fabulous library you've got there, Adam. All right, I just want to
Thank you, thank you.
let me clear up some stuff. And Jordan here. I can't tell. Jordan, you there? And that's some interesting stuff for him there. Oh, we'll wait a little bit. All right, so today we need to get through linkage, right? And so linkage is a very cool, very interesting problem, I guess, tool even in genetics, but it's also one that confuses the heck out of students. Oh, Rose, the answers should be available already. When did I have that? Because I extended the availability date. Oh, puppy's doing something. I think until, when was it, Monday night? I can't remember. And it should be available after that. Yeah, so that was 21st of June. Let me have a fiddle with that because it's always a bit of a mystery, to be honest. Rose, because some people can see them just fine, other people can't. So I will see if there's something that I've not checked or if something weird and wacky is going on, and I'll sort it out for you. Let me make myself a note. I had a really nice little notepad, but my wife tied it away wherever it's gone. Hey, do me a favor and grab me a notepad from the top of the fridge, would you? Notepad? Kind of feel around up there. There should be some notepads and stuff. I don't know why they always end up on top of the fridge. They just do. That'll do. Cool. Well, one sheet. Generous daughter. Yeah. But yeah, I'll check that out. Question about sex determination or sex determination or sex ratio.
The ratio. So I was reviewing the last slides from last class, and it says predicting the sex ratio, and you said it was a 1-to-1 ratio, and I was a bit confused on how you determine that.
So, oh, that's the other PowerPoint. Give me just a second, open that up. So it should be, just check and see what I've got in the PowerPoint. No, that didn't help. So if you have a homogametic and a heterogametic sex, so XX, XY in our case, then the homogametic sex, so the XX will always contribute an X chromosome to the next generation, because it's essentially kind of homozygous in a way, if you see what I mean, right? So there's only one possible type of chromosome that individual can contribute in the gametes. For a heterogametic sex, you have either an X or a Y chromosome in a gamete. And because there's no mechanism that I'm aware of which will bias that, it's kind of like essentially law of segregation, right, and law of independent assortment. There's no bias, so if you have two different possible options and there's nothing that will change or affect that which chromosome goes into which gamete, then you should have an equal chance of getting either an X chromosome or a Y chromosome in any given gamete from the male.
Okay, so the 1-to-1 ratio, one of them being homogametic and then the other one being heterogametic.
Not so much. I think about it more in terms of the gametes. Well, I mean, yes, but if you kind of wind it back a little bit, because the gametes from a male, in case of humans, will either have an X chromosome or a Y chromosome, like 50-50, then any potential offspring from that male and female should be 50-50 male or female in terms of probability. Obviously, that doesn't always work out. There's kind of weird, funky stuff that goes down through different families where you have just boys or just girls, right? And so that's something that I don't think we actually know why that happens. But if you have enough kids, like 50, let's say, and none of that funkiness exists in your family history, you should have roughly 25 and 25 of each sex. Cool. All right. Dang it, Jordan's there. I was super excited about talking about this. Well, anyway, I'm going to tell all you lot and he can catch up another time. So Jordan had a cool question about the urogenital development, males and females in humans. And so in humans, probably in mice and stuff as well. Let's just say humans. It's kind of easy to be definitive about it then. And I think it's 6 weeks gestation, give or take. You have both potential ductworks, essentially, right? They aren't the different reproductive organs yet because that takes time. And those are called the Mullerian ducts, which is essentially proto-female, and Wolffian ducts. Must be after some guy or woman called Wolf. And so essentially you have the two possibilities there, and in the absence of a male hormone or androgen, the Wolffian ducts degenerate, and in the presence of the anti-Mullerian hormone or Mullerian inhibiting substance, got a whole bunch of different names, the Mullerian ducts degenerate. So basically, up to I think about 8 weeks for definite, takes different levels of time, different species. You're both male, potentially male and female. It's only after that point, once the sex determining region Y transcription factor gets produced, that you start getting bias towards testes development and testosterone production, and then the proto-female ducts disappear and the proto-male ducts kind of continue development. And in females, you don't have that SRI transcription factor produced, so you don't get testosterone, you don't get anti-Mullerian hormone being produced, and so there's nothing to stop the Mullerian ducts developing, but there's also nothing to promote the Wolffian ducts from developing, so they degenerate. That's how you get that bifurcation in development based on the sexes, which is kind of cool. I love talking about development. And you also learn some really cool words. So the Mullerian ducts develop from the paramesonephros. How cool is that? Can't even spell it. Well, not easily anyway. Okay, so one thing that we had to catch up on is epistasis. That's a little bit of a hangover. Give me a second, I'll bring that up. And then get the chat back. Wherever it's gone. Yeah, well, there's even cooler words than that. Your kidneys develop, or at least part of them, developed from the metanephrogenic mesenchyme. That just takes too long to even type out. Don't worry about all those details, but they're just kind of fun to hear about. Yeah, development is really, really cool. It really is. It's such a fascinating topic. Okay. All right. So we're going to make sure we get through all of linkage today, ideally, because we've got a bunch of stuff to start on Tuesday. So I'm going to talk about this in kind of general terms because we don't go into it in huge detail in PowerPoint, but it is a very cool process, right? Something to talk about. So we've always kind of assumed that, you know, genes are independent, right? Not only are they inherited independently, but they act independently. And clearly that's nonsense, right? Because you don't just have a collection of proteins doing their random things on their own. You have a whole bunch of networks of, you know, metabolic pathways, developmental pathways, kind of steps in processes. And so it makes sense that, you know, if you have a whole chain of proteins doing different things, then you need all of those to work to see the outcome at the end, right? And a classic example of that is hair or fur color, right? So you have genes that are involved in pigment production, so melanin production. So that determines what kind of color you have. You know, whether you're have a dark, I always forget the words. I think it's eumelanin versus kind of lighter orangey red pheomelanin, I think it is. Pardon me. You also have many genes controlling the production of that. You have proteins which control the import of that pigment into the hair follicles or the fur follicles, right? And so if you have a mutation in any of those different genes, you're going to essentially affect the outcome of that entire pathway. And that's really what epistasis is all about. And there's an awful lot to epistasis, right? So I'm not going to try and cover all of it, right? Because I don't think that's really possible. You could pretty much do a whole lecture just on epistasis. But essentially the idea is that the kind of the alleles you have at one locus, or the genotype essentially, which is the collection of alleles you have at one locus, affects the phenotype that you express from a different locus, right? And so essentially epistasis means masking, right? So you're able to have the, you know, two different genes could be on different chromosomes, could be kind of genetically unlinked, right? So we're not talking about inheritance, we're talking about expression. And depending on what genotype you have at this locus, whether or not you see the phenotype at this locus really depends on the genotype at this one, right? And so give you another cool example. And there's lots of different types of epistasis. One of my favorites, obviously, cats. So a great example of epistasis, and this is dominant epistasis. You can get recessive epistasis too. It's fur color in cats, because cats are the bomb when it comes to genetics. So you have the white gene and I don't know what we'd call it. Don't know what its proper name is. And a pigment gene. So you have two alleles. It could be dominant allele equals black. Write this down for you. recessive allele equals, I think it's kind of ginger in cats, right? So you have two different genes, and essentially what happens is if you have I think it's homozygous, or if your cat, sorry, not you, obviously, because you're not a cat, has the white allele, your cat is white. Doesn't matter whether actually it's completely irrelevant. What the genotype for the pigment gene is. It makes no difference. You could be homozygous, or your cat could be homozygous dominant, or heterozygous, or homozygous recessive. It'll still be white. Completely irrelevant. So only your cat, sorry, I have to keep reminding myself. Do you see the phenotype for the pigment gene? So that's an example of dominant epistasis where one phenotype at one locus completely masks two possible phenotypes at another locus. Right. You can also have what's called recessive epistasis. And I wouldn't worry too much about dominant versus recessive because, again, we're not going into this in huge detail. But these are more just kind of examples to help you understand the process. And so recessive epistasis is really where you need a functioning copy of two or more different genes to see the outcome, right? And so these are the examples of these are typically in metabolic pathways, right? Where you have one enzyme that turns something from one thing into another, and then the second enzyme turns that something, that intermediate, into either another intermediate or the finished product. Right, so you need to have these things working in sequence to get what you need, in this case, purple pigment. So now if you're homozygous recessive for either of these genes, could be one or the other or both, it doesn't actually matter. But if either one of these doesn't work, the phenotype at the other locus will not be expressed, right? So even if enzyme P is completely functional, works gangbusters, that phenotype is completely masked by the fact that enzyme C doesn't work. Because if only one of them works, you're still not going to get that purple pigment. And so this is actually, remember we talked about complementation last class, where you can have mutations in different genes, and so you can actually complement them through a cross. Well, this is actually a good example of that, right? So you have two white, I think those are snapdragons, that are homozygous, right? So they're pure breeding. You cross them together and then you'll get a nice, I don't know, what's that, pink, I guess? Fuchsia? I'm not very good at colors. Light purple. You'll have function restored because you'll have heterozygote for both of those. Now, when you cross those together again, right, normally in Mendelian genetics and a dihybrid cross, you cross two heterozygotes together, you'll get a 9 to 3 to 3 to 1 ratio. In the case of epistasis, you will not. Depending on the type of epistasis, you'll get a weird ass ratio, right? And you'll basically see a limited number of phenotypes. So you'll get fewer than 4 different phenotype combinations for one, right? You can see there's only 2. It's either, oh, there you go, purple or white. And obviously the numbers aren't going to match. So if you ever see a F2 generation that produces fewer than 4 phenotype ratios, that tells you that epistasis is occurring. And the law of independent assortment does not apply. It has been broken. So for example, in dominant epistasis, you'll have a 12 to 3 to 1 ratio, for example. And there's all kinds of funky ones. Again, don't worry too much about it. You can read up on it if it kind of tweaks your curiosity. But one thing to be very clear of, and this is also something that kind of trips up students. Let me get the chat back, wherever it's gone. So do not confuse epistasis and linkage. So both will produce a funky F2 ratio, but epistasis basically equals a missing phenotype combination. Linkage equals, how would you put it? Fewer than expected non-parentals. Or if you put it another way, or an excess of parental phenotype. So in linkage, you still get the 4 different phenotype combinations, but they're no longer in a 9 to 3 to 3 to 1 ratio. In epistasis, apart from maybe some like weird and wacky ones that I'm not aware of off the top of my head, because I don't know all of the different types, you're going to be missing at least one of those 4 different phenotype combinations. So you'll have 3 or 2 different phenotype combinations. So don't confuse those two. They're very, very different, and a very different mechanism causes them. Epistasis is all about masking of another phenotype by the phenotype at a different locus. Linkage is all about 2 genes being physically close together and a lack of recombination.
So just a confirmation, when looking at the F2 generation, the very far end at the bottom right recessive, that's where the enzyme P is masked, correct?
Either P or C.
Oh, okay.
So, pardon me. So the only the flowers that are purple have to have at least one dominant allele for each gene. So if you kind of like look through these, you'll see with some that are homozygous, dominant, and heterozygous, and vice versa, some that are heterozygous for both. But you'll only see purple where there is a dominant allele for both genes in recessive epistasis. If you look at all the other ones, the other 7, the whites in that kind of that table, that Punnett square, every one of those has at least one of those genes, could be either C or P. It actually doesn't matter which. One of those is going to be homozygous recessive, or both of them, perhaps one on the kind of bottom right. So these would normally be the 3 to 3 to 1 part of that expected F2 ratio. But instead, they're all clumped together because they're all white. Even though they have different genotypes, they're still all white. Does that make sense? Yeah. Cool. Any questions on epistasis before we move on? It can actually be a pretty cool tool. It's something I use a bunch in my own research. And so you can actually use this to figure out whether or not genes are in the same pathway that you're studying and what order in that pathway they are. So it's a pretty cool tool. There's a lot more to it than that as well, as one would expect. Okay, so now we get on to linkage, right? So for those of you who are good at math, this is a walk in the park. For those of you that aren't, like me, it's a real mind bender. And so the thing to, and again, so I'm told, Dipper, so I'm told. There are like one or two maybe out there. Honestly though, if you're good at math and you like biology, you've got like the whole world is your lobster when it comes to jobs and research, because there are so many biologists because they're not good at math. So if you can do statistics and biology, then you're kind of made. In fact, Dr. Page, who taught this course before me, has made a very successful research career out of doing biostatistics for other people. So he's the one that does the modeling and analysis and various research projects for people. And so he gets his name on loads of papers because people can't do that stuff. I can't, for example. So, and he's actually now teaching the biostats course as well. So most likely at some point, you will need to take that and you'll most likely take it with him. So math is a pain in the ass, to be honest, sometimes, but as long as you understand the principles, then it's not so bad. So that aside, main point, exclamation, exclamation, exclamation. So hopefully you all pay attention. The recombination is random, and the probability of recombination is equal to the distance between two loci or genes. That is the fundamental key point that you have to understand to get linkage or understand linkage. So if you get, I don't know, let's say 1% probability of recombination when genes are, I don't know, let's pull a number out of my butt here, 100,000 base pairs apart. What's the probability when they're 1 million base pairs apart? Excuse the spelling mistakes. So if the probability of recombination occurring in a particular bit of DNA is random, and the probability of that recombination is equal to how far apart they are, if you increase the distance between those 2 points by a factor of 10, What do you increase the probability of recombination by? Yeah, you gotta be specific there, Rose. Because it's math, right? We can't just be a biologist and go, oh yeah, it's kind of like research. Got to be specific. I should have worn my biology is anything that moves t-shirt today. That would help. So if you have a 1% probability of recombination occurring in a chunk of DNA that's 100,000 base pairs, right, 100 kilobases long, you have the same probability in the 100 kilobases next to it, and next to it, and next to it, and either side. So every 100 kilobase, or that 100,000 base pairs, you have a 1% probability of recombination occurring in that chunk. So if you have 1% probability, kind of going the wrong way. If you have 1% probability in 100 kb, what's your probability of recombination when in 10 lots of those that you have recombination there occurring once in 10 lots of 100 kb? The probability is 1% in one lot. What's 1 times 10? 10%. Okay, so what about if those points are, and I'm not sure if this is, I'm just making numbers up here to illustrate a point. What if those 2 points are now 10 million base pairs apart, right? So 100 times the distance of the 100 kb, where your probability is 1%. So the points aren't just 10 times further apart now. To get 10% probability, they're 100 times further apart. 100%, exactly. That is the main point. What if they're 50 times apart? 50%. You know, that's what matters. So the probability of recombination is exactly proportional to how far apart those 2 points are. The closer they are, the lower the probability. The further they are, the higher the probability. Because the probability of recombination occurring is random. Could occur here, or here, or here, or here, or here, or wherever, right? It's not 100% random because different parts of the chromosome don't behave exactly the same way. But for our purposes, it's random. So the further apart something is, the higher the probability of that random event occurring. It's like those really annoying games that are like carnivals and Six Flags. You know, we have those bottles and you're supposed to throw rings into them, right? The more bottles you have, in theory, the higher the probability that you're going to get a ring into them. Oh, sure. That's exactly right, Rose. The distance between two loci is directly proportional, if I can write that, to the probability of recombination occurring between them. And it's because it's random, right? There's nothing direct in recombination. And so another point that's really important is that recombination always occurs in meiosis, has to, right? Because it's what holds those homologous pairs together in, well, basically until anaphase 1, right? Those kind of swapped over bits that stick to the other chromatids. What we care about is whether or not recombination, thank you, autocorrect, is occurring between the two genes we're looking at. So recombination is always occurring, right? It's not that it just doesn't occur. But if you're looking at recombination between 2 points, the probability of that happening, and again, this is 2 points on the same chromosome, right? Put that down just in case. The probability of that occurring is going to increase the further apart they are. Or another analogy, I don't know if any of you played football or soccer as they call it, or soccer or football as they call it. You know, at school or whatever, when I was a kid, we always used to just use like someone's sweater or t-shirt or whatever as the goalposts. And always like some goalies on one of the teams would kind of try, you know, pushing those goalposts closer together, right? Because it's harder to score the smaller the distance between the goalposts, right? Random probability if you're not kind of like looking at where you're going. And it's exactly the same deal. Further apart, two things are, the larger the probability that you get a random event occurring between them. And that's a super, super central point to all of this. Without understanding that, none of this is going to make any sense.
Just a curious question, but like, does the probability of recombination happen at the end of meiosis or throughout those different phases?
It's purely based on recombination, which occurs in prophase 1. Because that's where the homologous chromosomes kind of pair and line up using the synaptonemal complex, and you get the crossing over occurring. Everything after that is just kind of like sorting the various chromosomes and chromatids out. Okay, so everybody got that kind of main point. If you don't, just ask, because I want to catch any misunderstandings or lack of understandings before we get further, because otherwise you're going to get lost. Okay, Valeria, that's cool. It's good to hear that because I don't want you to be confused. What bits kind of stick? What's kind of sticking in your throat, so to speak? So I can figure out another way of kind of explaining that might catch better. I don't want to rely too heavily on sports analogies because I'm not much of a sportsman. Ah, okay. Cool. Thank you, Marissa. Hopefully this will help the others too, right? So what we're looking at is a random event. And in this case, it's recombination or synapses or crossing over, whatever you want to call it. So it's basically is the same for any given part of the chromosome. And that there are exceptions to that, which we'll get to later on. For every given same size chunk. Right. Sorry, dog, I'm gonna have to put my foot down. Oh, she doesn't mind, she's asleep. Yeah, where is she at? Can you see her? There we go. Doggo number 1. Now she's gone off. And also puppy toy, just in case she ends up here and starts chewing on my feet. So often these things are easier to draw than they are to explain. So let's say you have a chromosome. And you have a chunk about that big. So the recombination might occur here. Right, so if you have your your homologous pair, like so, give or take. Make sure we get it all straight. And you'll end up getting recombination between these two non-sister chromatids here. Right? Now, if it occurs here, it's not going to occur here, or here, or here, or here, or here, or here. So you only get one recombination per arm of the chromosome. So if you have two genes either side of that, you will see a different combination of alleles, presuming they're heterozygous. It's heterozygous. You'll see a different combination of alleles in the offspring. So you'll get, essentially, if you have recombination there, you'll get and the two parentals, like so. And you'll get two recombinants. All right, that's the kind of basic principle of recombination that we learned when we talked about this in meiosis. sorry, I have to stretch my leg out, otherwise stuff happens. Okay, so
Wait, I wasn't done. So
oh shoot, I'm
for
sorry.
sorry, no, for the last chromatid at the very bottom, was it capital A, capital B? For
So
the
essentially, if you have, what did I use? There we go.
because you had the cross where it was AB, AB, little a, little b, and then I want to say it was big A, little big A, or big A, big B.
Okay, so essentially what you have is it has to be a heterozygote because otherwise you're not going to see any difference. Yes, thank you, Marissa. So on this chromosome is going to be, or chromatids. Both of those chromatids are going to be AB, right? Because remember, these are carbon copies of each other, right? They're not different chromosomes. This black one is one chromosome. You just got two copies of it, two copies. Now, if the other chromosome has to be the opposite alleles, right? So we have a heterozygous individual. So these are called the parental chromosomes. And they're called parentals because, well, that's what this individual got from their parents. Now, if we have those two genes lined up like so, and recombination occurs between them, right? Because you can also have recombination occur like here. If recombination occurs here, you'll get exactly the same genotypes in the gametes. What you care about is recombination occurring between these two genes? That's what really matters. That's what you'll see, right? Because that will generate new combinations of gametes. So if recombination occurs between those two genes, what you'll see is this chromosome up here, well, currently a chromatid, but after, you know, meiosis does its work, it will be a chromosome. You'll see the dominant, you'll see the recessive, right? Those are the parentals, but you'll also see new combinations, right? You'll see a dominant A and a recessive B. And the inverse, like the mirror image, lowercase a, capital B. Ah, that wasn't very good. These are, as Marissa put it, oh no, gone behind the couch cushion there. These are the recombinants because these are different to what you originally had in that individual. Yep. Now, this is where we get into the math. If the, and I'm just gonna, let's write this down first, right? So let's say probability is 1% of that occurring in that given kind of bit of DNA or length of DNA, whatever you want to call it. Now, if that's the case, probability is 1%, you'll get 1% recombinance and 49% parentals. Actually, that's wrong. 99%. Confusing myself here. Because only 1% of the time will that recombination event occur between these two genes, if they're that distance apart. So you'll end up with 1% of your gametes, and therefore the offspring, being recombinant, having a different combination of alleles than the parents. Okay, so hold that in your head. I'm going to take a picture of this. My camera go. And we'll change things up a little bit. What's my green pen? Does that work? No, green pen does not work. What about this one? Can you see that? Yeah, kind of. Yeah, that's all I've got for colors. I have to pick some more up when I go into the office. Okay, so now let's look at a different combination. Right. Now we're looking at two different genes. Oh, crabs. Let's not use C. C absolutely sucks for letters. Okay. These genes, and this is kind of exaggerated, these genes are now 3 times further apart than A and B were. So 3 units of distance, let's say. So now we have 3 times the probability that recombination will occur between these two genes. What's the probability of recombination now? If one of these units is 1%, they're now 3 times further apart. Yay, 3%. Now you're 3 times more likely to have a recombination event, right? And now that could be here, right? It could be here. It could be here. Doesn't matter. As long as it's between these two genes, you'll end up seeing more recombinants. And so now you'll get the same deal. You're going to have the parental. Give me a second, Rose. Let me just follow through on this, and then I'll get to that. You'll have your parentals, you have your recombinants, different gene letters because we're talking about different genes. Now you have 3 times as many recombinants because the genes are 3 times further apart. And so you'll have 97% parentals and 3% recombinants. What about if you're actually looking at genes A and E, right? Which are 4 times further apart. What's the probability of recombination now between A and E? It's going to be 4, right? So it's directly proportional, at least in the arms of the chromosome. The further apart they are, the higher the probability that something will happen. So another analogy, Rose. I don't know about you, but I'm absolutely a terrible shot at getting stuff in the trash can. So how much easier is it to throw something into a trash can that's the size of a, oh, you don't have dustbins here, like one of those big things that you put out on the curb versus, you know, one of those little trash cans you have underneath your desk from the same distance, same size of object. Which is the easier one to get that object into?
Could you put it back actually?
So what again?
Could you put it back on the board again? Sorry, I was still writing.
Oh, I'm sorry. Now, I'm not talking about how far away the trash cans are, Rose, but how big they are, how wide the aperture. The bigger one, right? If you were, and I wouldn't recommend this, to get a bow and arrow and a archery target and close your eyes, again, not a good idea. Would you be more likely to hit within the main ring, or would you be more likely to hit a bullseye? Again, with your eyes shut. Yeah, obviously you're not shooting straight up in the air. You have some kind of random guidance, I guess. Like someone points you in the right direction. Right, it's going to be the main ring. It's going to be the bigger thing. And that's the point. This is a random event. Recombination is not guided. So you have essentially along the whole chromosome arm, you have 100% chance of recombination. Every round of meiosis, you will have recombination somewhere in here. Guaranteed, without a doubt. Now, when you start shrinking that size, right, the probability of recombination in that bit between my fingers gets smaller the closer my fingers are together. Just like your chance of hitting the bullseye is smaller than the chance of just hitting the target if you have your eyes shut. Or even if you have your eyes open, actually, depending on how good of an archer you are. So if the two things that you're looking at, say genes A and D, yeah, exactly. That's exact. I mean, I didn't use darts because I didn't know how many people play darts. I played it when I was a kid growing up. That's because I lived in a pub. So yes to Rose, the further apart the two things you're interested in, the more likely that random event will occur between them. the closer they are together, it's like hitting the kind of the, what is it, the 25-point ring on a dartboard, just that one, or the double or the triple or whatever. It's much, much harder. It's much less likely. Now, Richard, this isn't, or Adam, I don't know which you prefer to be called by. This isn't about how many alleles you have, right? We're only concerned with, at this case, 2 different loci genes. Now, obviously, you have loci all the way along this chromosome. You have different genes all the way along. But you're only concerned about the frequency of recombination between, at any given time, 2 of them, at least until we get into the triple point crosses. So the further further apart those 2 are, yeah, that's not a perfect analogy, Valeria, because it's not random. It's kind of along the right lines, but it's really just random probability. Further apart things are, the more likely that random event will occur between them, right? So the closer they are, right, trying to fit my finger between, not going to happen. Further those fingers get apart, the easier, more likely it is something's going to pass between them. And that's the deal with recombination. Okay, I'm going to leave it there for now so we can rest our tired brains and stretch our squished butts. Oh, cool, good. Sometimes it just takes talking about things in a different way for it to twig. Not everybody catches it in the same way. All right, I'm going to go stretch my legs, and we'll pick this up when I get back, and we'll have a load of practice problems to go through, right? Because that's really how you get this. At least in my, oh, Dan didn't stop the share so you could see the board. Sorry about that, I forget. See you in 15 minutes. Go get a coffee.